today, and we will get started. It is my very great pleasure to welcome the first speaker of our 2025 Catalysis Hub webinars, Joelle Pelletier. Sorry, my French accent is atrocious, so apologies if I pronounce that wrong. From the University of Montreal, and she's going to talk about accelerating the discovery of substrate promiscuity in biocatalyzed oxidations. Thank you for talking to us, Joelle, and please take it away.
Thank you very much. Thanks everyone for being there, and it's a pleasure to be a guest at the UK Catalysis Hub. So I will start presentation. Is everything good?
Yes, we can see your slides.
Wonderful, thank you. Okay, so today I'll be sharing with you some of the work that we've been doing over the past quite a few years, working in particular with the enzyme cytochrome P450 BM3, and in our ways of thinking of how we can modify and improve biocatalysts to diversify the reactions that we can address. So, first question, why use biocatalysts, or why not use them? So an example that comes to mind immediately is this publication in 2017 by Merck, which presented the old path to the synthesis of their antiviral islatravir, which you can see up here, many steps, many reagents, several steps that are not particularly green. And they presented a new approach. And in this approach, all the colored boxes are enzymes. And each of these enzymes has been engineered because in its native or wild type form, each of those enzymes would not efficiently catalyze the desired reaction. So by this way forward, they were able to massively improve their process, improving their overall yield, and they were able to do this in a continuous flow process in water with no organic co-solvent. Since 2017, since that publication, they've continued to improve the steps by adding other tweaks in the system. And so this just shows that the industry adoption of biocatalysis. So why biocatalysis? Of course, enzymes are derived from living systems. Of course, they're biodegradable. These are good things, so they're not particularly harmful for the environment. They tend to offer high selectivity. We can think of chemoselectivity, regioselectivity, and enantioselectivity. They can offer direct synthesis routes, meaning that we can often avoid protection groups and therefore deprotection steps also. They generally work in mild aqueous conditions. Again, this is green. And so overall, they tend to provide sustainable routes to synthesis. But of course, they come with challenges. Enzymes tend to have limited stability. And this is something that has to be addressed. They may have low activity for a desired reaction, and they may have a narrow substrate scope. Nature doesn't do all the reactions that we want to do industrially. So we need to be able to tailor enzymes to meet our synthetic needs. So I'll dive right away into the main topic, which is to catalyze oxidation reactions, and I'll focus on cytochrome P450 oxidases. So these enzymes, they are monooxygenases, and they have been dubbed the most important oxygenase class for organic synthesis. And I'm showing you a few different reactions here, which are very useful. The formation of a styrene oxide here, or another epoxidation shown here. The advantages are that you can use mild conditions for this direct hydroxylation of also a benzene to hydroquinone, for example. This cannot be done by conventional chemistry. The enzyme, the cytochrome P450 oxidases, they have a heme group, and so there's heme iron, and this reactivity is actually happening on the iron. So in a way, it's organometallic chemistry. So they function by using electrons. This is a reductive oxidation reaction. The electrons come from the natural cofactor, NADPH, and use oxygen directly from air. So quite simple to operate in some sense. However, you will notice that in the reactions that are shown here, there's wording underneath the acronym for the enzyme, P450 BM3, as I will refer to it. And these are mutations or substitutions that have been made to the enzyme in order for the enzyme to be able to conduct these reactions, to conduct them appropriately. And that brings us to the question of exploring functional sequence space of enzymes. Enzymes are big catalysts. They are made up of many amino acids, and these can be diversified. They are diversified in nature. In nature holds many variants that we can go look for, but then we have to test them. And so it's this, how do we balance our search through sequence diversity in order to attain function, the desired function? So eventually where we want to go with this is we want to have enough experimental input to inform computational algorithms. And these computational algorithms eventually will aid in the design of new reactivities. So of course, it takes a lot of time to obtain experimental input, but there's somewhere where we can start, and that's looking in the literature. So postdoc Doug Fancher in my group, he spearheaded an effort to scour the literature for everything that had been published about engineered P450 BM3, so this particular cytochrome P450, all the reactions where variants have been reported to catalyze a reaction. So he scoured through aromatic hydroxylations, drug or drug-like molecule hydroxylations. We can also find hydroxylation of native-like substrates. The native reaction is to hydroxylate long alkyl chains, but short alkyl chains also can be hydroxylated by engineered variants. Epoxidation, aromatic epoxidation can occur, alkyl epoxidation. So of course, the epoxidation is always on the same group, but the R groups can be different. Carbene transfer has been engineered in for cyclopropionation. This was along with this next reaction, the nitrene transfer for aziridination. These two reactions were among the Nobel-winning reactions of Francis Arnold in 2018 for the directed evolution, and there's other reactions too. So all these reactions can be catalyzed by variants of cytochrome P450s. Now, what are the mutations or the substitutions that give rise to these different reactions? So if we look first at aromatic hydroxylation, in red spheres, I'm illustrating all the residues that have been associated with literature reports of new aromatic hydroxylation, new or improved. And then if we go to drug and drug-like hydroxylation, these residues differ. And they differ again for the reaction on short alkyl chains, long alkyl chains, aromatic epoxidation on alkyl, cyclopropionation, carbene, nitrene transfer. So we have this mass of information. But if we want to enable further discovery, if we want to enable machine learning, for example, then the information has to be in a searchable format. So in order to do this, Doug Fancher built a searchable database. So in this database, all the different variants of cytochrome P450 BM3 scroll down a list, and the different categories of reactions are shown, and we can scroll down. And so this database is searchable. It's called the P450 BM3 variant database, and it catalogs many variants over the 9 reaction types I've shown you, over 350 references, many entries. So what does it look like? each line gives you one variant and the reaction classes that it can address. We have the parent enzyme, which typically is the native or wild-type cytochrome BM3 and the variant that we are looking at. But sometimes there's so much literature that there are some variants that have become anchored in the literature and can then serve as parents that we further vary. And so we can refer to some different parents. And a lot of information is given. And the key is that you can automatically sort and filter, apply your filters, you have options, and it automatically redirects you to the reference paper so you can get more details. So we're working on getting this online for users. And if anybody's interested, please do contact us, because we have already given access to this database to a number of people. And it's a fully downloadable database, so you can use it on your own also. So we have now this capacity to screen through this database. And what can it do? For one thing, it can we can use it to look for gaps. So if there's a gap, then we will be able to then expand the substrate scope, for example, of this library. It can then give you an idea of a starting point, so a variant for an activity of interest that you want to delve into deeper. You can also use some variants and combine them to create a combinatorial library and go further towards certain reactions. And what we're working on is to use this massive information for computational prediction of potential activity. And this, it's such a wealth of information that we hope that it will help bring machine learning farther for to help us in evolution of enzyme activities. So one thing we could do directly with this database is bulk data analysis. And one of the things that we found is the positions that had the highest sequence diversity for improved specific reactivity. And so what we mean by this is at any given position, what was tolerated, how many different amino acids could be placed at a single position to give rise to improved activity. So in that case, these were positions that could hold 7 or more among the 19 possible different amino acids that one can use, since there are 20 amino acids. And what we saw, if this is highlighted here with the blue spheres, is that enzyme engineering has been successful in positions that are highly variable, but also at highly conserved positions. And this means highly variable and highly conserved in nature. So even though nature has conserved these positions that are marked in red, we can still modify them in the lab, engineer them, and expand reactivity. So we should not necessarily avoid mutating positions that are highly conserved when we're looking for new reactivity. Here, the substrate is shown bound in, and this around the substrate area is what were the most frequently reported in the literature. And this makes sense because that's where you're really changing what's happening with your substrate scope. So at this point, I'll embark into our experimental work, which is to engineer cytochrome P450 BM3 to produce indigo. So the native enzyme does not catalyze or hardly catalyzes this reaction, which is to hydroxylate indole, sorry, not hydroxylate, to oxidize indole to indoxyl, and then this spontaneously air oxidizes to dimerize it into indigo. Again, the native enzyme does not or hardly catalyzes this reaction, but this reaction is improved by a number of variants. And so there are references here. The group of Turner and Flitch had reported this a number of years ago, 2005, for example. And what they noticed, the reason for which this is interesting, is that they and other authors who are cited here noticed that variants that produced indigo tended to hydroxylate other non-native aromatic substrates that are shown here with higher efficiency. So there seemed to be a correlation between the capacity to oxidize indole and the capacity to hydroxylate these different molecules. So when we began this work, there were point substituted variants at 6 different positions that were known to produce indigo. And our objective was first to question, can we broaden this library of indigo positive singly substituted variants? And if so, then we could verify the hypothesis of whether more indigo positive variants are predictive of even broader reactivity. And the reason for which indigo is such an interesting, the indigo formation is such an interesting reaction to look at, is that here you're looking at a rectangular Petri dish, and it holds 96 different colonies of E. coli, and each colony is expressing a different variant of P450 BM3, and you can easily see which ones are producing indigo. So indigo is just so easy to screen for that if it allows us to focus in on variants that are more likely to then catalyze an oxidation reaction of interest, well, this would be a winning game. So this should be easy because, enzymes, they're pretty easy to engineer. Well, not really. So cytochrome P450 BM3, here I'm illustrating the catalytic domain. It holds over 400 amino acids, and illustrated are the tunnels that lead to the active site. The active site is illustrated here by the heme molecule. So the activity happens just above this heme molecule, and all the tunnels that are leading in to the active site. Now, this is actually not all, because this is one of the 3 domains. Function requires 2 further domains that are not shown. And it's even more complicated than that, because this enzyme functions as what is called a domain-swapped dimer. So we need 2 enzyme molecules, and they are kind of wrapped together such that the catalytic domain of the one monomer needs the 2 further domains from the other monomer in order to be functional. And the mechanism is complex for these reasons and for many other reasons. So to engineer this is quite complicated. That's why we need some high-throughput methods to help streamline the process. So again, what we want to do is to see whether we can expand the library of indigo-producing variants. So we did this by performing at first site saturation mutagenesis, so mutating to the 19 different possibilities. And here you have the active site heme. We're looking at the active site just above the heme, and that's where we selected these amino acids to mutate, and we're screening again for indigo formation in E. coli that expresses our variants. So this was successful, and it encouraged us because we found a number of new indigo positive variants. And we also looked at whether this was related to increased substrate promiscuity by looking at a new reaction. So this new reaction is the oxidation of 4-phenyl-2-butanone. And the product it gives, raspberry ketone, is very high value. So this is natural raspberry flavor. It's very expensive because to extract it from raspberries is apparently a complicated process. And it would be great if we could make this from a naturally sourced 4-phenyl-2-butanone, which is possible, and then to biocatalyze the product. So this would be kind of a natural flavor. So when we identified our new indigo producers, we found that those that produced raspberry ketone were indeed indigo producers, and any variant that did not produce indigo did not produce raspberry ketone. So this small-scale effort encouraged us to continue. Again, this is the active site of cytochrome P450 BM3. and in red is the heme. So above the heme is the active site cavity, and we mutated 42 different amino acids, other than the 5 that we previously mutated, that are within 20 angstroms, any atom being within 20 angstroms of the heme, and to the 20 possibilities. And then, of course, we can pick the colonies where indigo is produced. And then next generation DNA sequencing allowed us to identify the mutations that produce indigo or don't produce indigo. And finally, we can quantify the indigo production. So it gave us many new point substituted variants that produce indigo. We found nearly 100 that produce indigo. And this is shown in this graph where we have the 20 possibilities of amino acids, and these are the 42 mutated positions. And in the darkest blue, this were the top producers of indigo. The light blue, weak producers of indigo. In this kind of beige are those variants that we identified that did not produce indigo. In white are the positions where we simply did not find those variants at all. And the X's, black X's, indicate the native sequence. Of course, the native does not produce indigo in any useful form. So we found about 100 new indigo producers, and these include 18 previously unreported positions. So that answers one question. Yes, we can find many indigo forming variants. So if we look at the 18 previously unreported positions, they are shown in blue here, whereas in beige are positions where we did not identify any substitution that gave rise to indigo formation. And if we now look at just our top 32 indigo producers, they are all located within 13 different positions. And you see that we're kind of shifting to the left of the active site. We would be interested in better understanding why these substitutions give rise to indigo. That's an ongoing effort. So again, here's our target reaction, this raspberry ketone synthesis. And our question now is, does the indigo formation by this variety, this much greater variety of P450 BM3 variants, does it correlate with increased substrate promiscuity? So will we be able to identify more of these raspberry ketone producers? And we are monitoring using non-native standards also. We're looking for the oxidation of anisole and the oxidation of naphthalene. So the workflow is such that we have a particular target substrate, and we have created our catalyst library, and we can screen using indigo formation, but now we're looking for hydroxylation of anisole and naphthalene. I'll get back to this. Using our representative substrate, we identify our active catalysts, and we characterize reactivity. So what we need here is we need a screen that is high throughput. And I'll just mention now that an advantage of doing this is that our catalyst library that we synthesize can be rescreened afterwards, and I'll get back to this. So here's the colorimetric screen that we're using, where in looking for increased substrate promiscuity, we are looking for hydroxylation of anisole and naphthalene, or of synthesis of our raspberry ketone. And we're using this assay that was reported by Schwanenberg, and that uses 4-amino antipyrine and reacts with phenol to give this colored compound. So the reaction is effective with phenol, and a question mark was how substituents on our phenols, so once we have, for example, hydroxylated anisole, hydroxylated naphthalene, would these still be reactive with the amino antipyrine to give a colored product? So happily, the answer is yes. And we were able to, therefore, apply this assay to identify our products of hydroxylation. So following hydroxylation, we can identify the products. And for anisole, what we see is that the indigo positive variants here are much more successful on average, so statistically, relative to the indigo negative variants. And this is also true for naphthalene. And in particular, this does not relate to any difference in expression. Again, this is statistically. So it is actually reactivity that is changing. And we also note that all the top variants are indigo positive. And this is what interests us, most likely, those that are the most reactive. So if we look at the correlation, here we're correlating the production, the hydroxylation of anisole, the hydroxylation of naphthalene, and we see a certain correlation, which we can also cross-correlate with the production of indigo, which is shown in color. So what we see is that if we look only at our top indigo producers, some are not effective at hydroxylating anisole or naphthalene, and some variants that do not produce indigo are effective in hydroxylating anisole and naphthalene. But overall, if we focus, for example, on the top producers of indigo only, then our top producers of indigo are those that are the most effective overall in this substrate promiscuity, in these promiscuous reactions. So indigo generally correlates with oxidation of these aromatic molecules. And we can also go back to our target reaction. And again, we have a loose correlation where if we target the indigo producers, then we will more effectively identify increased reactivity for this hydroxylation of 4-phenyl-2-butanone. So at this point, we again can look at a number of substrates. So let's see if we can broaden to the prediction of more broadly aromatic hydroxylation. And at this point, what we can consider it is an all against all screening, meaning that we will screen our catalyst library against a panel of target substrates. So again, we are using our reporter assay. Our reporter assay will monitor whether hydroxylation of an aromatic compound has occurred. And this is the panel of compounds that we screen. And what you can see here, for example, for our first compound here, we can see that two different products of hydroxylation do give a signal in this reaction. The colors are different, but we can observe product formation. And this is, again, compound 11 here. So for each of the compounds, a panel of products formed with the amino antipyrine reaction were monitored using standards. And so when we screen this, I will show you one example here with compound 5, which gives, so this chlorobenzene. We can see that the indigo positive variants quickly, at a glance, give a higher hit rate than the indigo negative variants. And the indigo positive phenotype not only gets more hits, but also gives increased aromatic hydroxylation. So we are repeating this with our panel of compounds. And again, we can rescreen. And what we can also do is to identify new reactivity. So the reaction that's of interest here is to look for styrene epoxidation. So styrene epoxide can be monitored using a colorimetric screen, here using nitrobenzylpyridine, which gives a blue-purple signal. And this colorimetric screen for styrene epoxidation, we have now applied. We've begun this with styrene, and we see that we get a much higher hit rate for the product of styrene oxidation. So the styrene epoxide gives a much higher hit rate with the indigo positive than the indigo negative point substituted variants. So what is ongoing is the characterization of conversion, regioselectivity, and enantioselectivity of the top variants. And sorry, we are screening substituted styrenes. So this is a panel using the wild type cytochrome P450 BM3 and a panel of substituted styrenes. We see that the wild type enzyme can give rise to epoxidation of different substituted styrenes, but a particular variant gives reactivity towards a greater number of substituted styrenes and gives a greater extent of reactivity. Sorry, these are the compounds that were screened, the different styrenes. And so we can generate now a table where we have, as the horizontal, we have our different compounds, our different substituted styrenes that are screened. And in the vertical axis, we have our variants, our P450 BM3 variants that are screened. And so this is actually a massive table because we are screening hundreds of variants at a time using this very rapid screen. So this is actually ongoing now. So what we know now is that indigo formation, so the oxidation of indole to form indigo, correlates in general with a higher hit rate for hydroxylation of a variety of aromatic compounds, epoxidation of substituted styrenes. And so where we would want to go with this through our workflow, where we can screen rapidly, is to characterize reactivity and eventually move toward reaction cascades, scale the reactions, and eventually transfer the knowledge. Now, we can also see this as iterative processes. I've been speaking up till now about point substituted libraries. So all the results I've shown are where a single amino acid had been changed in the target enzyme cytochrome P450 BM3. But we can now recombine those substitutions, and in an iterative process, we can now combine substitutions and see what that gives us. So the hypothesis here is that if we combine indigo positive substitutions, we will enhance, we will broaden perhaps promiscuity towards hydroxylation of aromatics as one target. So up till now, what I have showed you is we started with the native P450 BM3, and we performed site saturation mutagenesis at over 40 positions. We obtained a library of singly substituted variants, which were either indigo positive or indigo negative. That's what I showed up till now. So what we then did is we picked out from 15 positions, we picked out 56 different substituents. So for example, one position can hold 4 or 6 different amino acids that are indigo positive. So we combined 56 variants at the positions, 15 positions that are shown here. They're color-coded according to the element of secondary structure where they belong. And these were recombined. Twist was our partner in doing this to give a multiply substituted variant library. And so we have a few tens of thousands of variants theoretically that each include between 2 and 9 substitutions, on average about 3.5 per variant. And my bet was wrong. If you're going to be combining indigo positive substitutions, you'd think that all the products, all the combined variants would be indigo positive. And they're not. Really, I'm glad I didn't bet on this because I would have lost money. So only about 33% of the library is indigo positive. And I'm not quite sure yet what that is telling us, but that's just a fact. So now the question is, if you take multiple indigo positive substitutions, combine them, some of them give indigo positive phenotype, some don't, but they're all based on the indigo positive substitutions. How will this pan out with respect to expanding promiscuity? So again, we're back with our colorimetric assay to reveal aromatic hydroxylation. Does the combination of these substitutions enhance aromatic hydroxylation? And again, here's our panel of 12 compounds, so differently substituted benzenes. So here what we have as combination is about 5,000 variant screened per compound combinations. This is what we actually did screen. So I'll show you this graph here. On the horizontal axis are our 12 aromatic compounds. We are looking whether they are hydroxylated. And if we look at the wild type enzyme, the wild type doesn't do much on any of these compounds except for compound 12, which is this dichlorobenzene here. But nothing much happens with all the other compounds. So all our variants, almost all variants, increase the activity. And so the combination of indigo positive substitutions really can increase the activity relative to the native enzyme. However, we did not see any statistical correlation between the combinatorial indigo positive and indigo negative variants. But the most active variants that we can see here are typically indigo positive, though not always. So again, what we have to remember is that all the substitutions within this library all started as indigo positive single substituents. So if we look, for example, for compound 1, we have just ranked as in their order, increasing and then decreasing in reactivity. We have a kind of a similar distribution. The indigo positive variants seem to be generally more successful. They don't drop down as fast, but the indigo negative variants certainly hold some gems. And this was done as initial rates. And again, we have to remember that all these indigo negative variants shown here are derived from indigo positive substitution. So we still do have a good success rate. And again, the native enzyme doesn't catalyze this reaction. So what about looking at longer reaction time with NADPH recycling? Can this actually be of any use? So can we look at phenol production by engineered cytochrome P450s? So this is our dichlorobenzene here, which becomes dichlorophenol. And one particular point substituted variant was particularly successful in producing the compound 12C. But if we look at through our panel, we see that we can also get some variation in the product scope. So indeed, we see that screening for indigo positive variants as indigo positive as either point substituents or in the context of multiple substituents is an efficient strategy for diversifying product formation. So this brings me back to our perspectives, which is to feed this information for machine learning. And what we want is to leverage our experimental data sets, which are very rich for machine learning. We want to use our experimental data sets to train models for prediction. We have both the single variant databases, we have the combinatorial libraries, and then we can generate additional features using the structure. So I showed you at the very beginning the tunnels, and there are elements of secondary structure also that we can look at, and we can encode mutations eventually. So this is a perspective that we are working toward. Now, in the meantime, another perspective that we are working toward is flow chemistry. Cytochrome P450 could be interesting for flow chemistry, except for an important observation that when P450 BM3 is used in solution, for example, if it's used in lysate of E. coli or as a purified form, it rapidly loses its activity. And however, once it's immobilized, it can be highly stabilized. And so we looked into this. But another challenge for using P450 BM3 in flow chemistry is the cost of using NADPH. And recycling is also costly because we're flowing everything out of the system constantly. So an alternative source of electrons to NADPH could be to use photochemistry. So in this case, what we would do is to use a sacrificial electron donor that is low cost and a photoactivated electron transfer mediator, again, low cost, in order to reduce our substrate, and with the flow of air would give rise to the oxidized product. So to do this, we built an in-house photobioreactor. So this is the actual cell in which we load our immobilized enzyme. This is blue because it's immobilized on nickel NTA or cobalt NTA or whichever. And this cassette fits into a device that is topped with a light source. And the top plate of this cell, it's a flat cell, is glass. So we can use this as a photoreactor, photo flow. And we looked at the conversion of lauric acid, which is a substitute of a native substrate of P450 BM3. And we see that with a residence time of 30 seconds, we can get essentially complete conversion. And this conversion is stable at room temperature over days. And the one disadvantage, however, is that this reaction was only at 20% of the rate of using NADPH as an electron source. So the use of the light was not as effective. Nonetheless, we are able to get full conversion with a rapid residence time. And so ongoing work is to convert this into a fluidized bed reactor. And the reason is that the solubility of oxygen in the aqueous media is, by physics, limited to a relatively low concentration. And it is therefore spent quite rapidly. And that's really the limiting factor. So in this fluidized bed reactor, which is a fancy name for a column, we bubble air to keep the oxygen level at its soluble maximum, and we sheath the column with a light source so that we can perform the photo flow. So that's what I wanted to tell you today, give you some ideas of what we have done with cytochrome P450 BM3. We're thinking of how we can expand its substrate scope, how we can expand its scope of reactivity, how we can do this efficiently. So using colorimetric screens, whether it's indigo formation, whether it's the coupled assays to detect product formation, and hopefully working towards flow catalysis eventually. So I tried to mention or show photos of the people who did the work, and I'd like to thank my entire group. These are the people who contributed directly to the P450 work over time and sources of funding. A couple of papers that I highlighted during the talk. Here we are standing in front of our brand new building. This photo was obviously taken in summer. I asked them for a winter photo, but somehow, for some reason, nobody came. Thank you very much for your attention. I'll be happy to take any questions.
Many thanks for a fascinating talk. It's the worst thing I think about webinars if we can't give the speakers a round of applause for the excellent talk. But thank you very much.
I'm not hearing you very clearly.
Sorry. Is that the
It's still garbled. Maybe if I stop screen sharing, maybe we can get back just on the perhaps this will be better.
Yeah, it's fine. It's obviously just mine that's.
Okay, it's a bit garbled, but hopefully I'll be able to hear. And can you hear me clearly?
Yes.
Okay. Oh, I'll listen.
So I would say thank you very much for an excellent talk. And it's one of the downsides of webinars that we can't give you a round of applause from all the attendees. We have had a question come in already, which is how does the workflow chooses the site mutation? Is it just random, and does it account for the electrostatics?
So the workflow at this point did not include any specifics except for position, a proximity of the positions near the active site heme. So basically the iron on the heme was our center point from which we decided which mutations to make. And from there, observing indigo phenotype was how we went forward. So we did not look at, and this was agnostic. So we simply went for a full site saturation mutagenesis at each position. Mind you, that was the intent. In practice, we did not obtain a representation of all 19 substitutions at each of the selected amino acids, just because that's technically how it happened. I hope, does that answer the question?
I'm afraid I'm an engineer by training, so I'm more hit it with a hammer than biological systems. But we've got another question come in. It was interesting to see that immobilized enzymes have a longer stability than enzymes in solution. Can sufficient amounts of enzymes be immobilized so that substantial quantities of products can be formed?
I missed the second sentence, please.
Can sufficient amount of enzymes be immobilized so that substantial quantities of product can be formed?
Yes, thank you. Yes, it is interesting. And this observation of stabilization through immobilization is not new. It's not necessarily universal, but it has been seen before. In some cases, different materials for immobilization will give different results. So you may have to screen different types of immobilization. And is it possible to produce enough enzyme? Yes. I mean, depending on, what your application is, we have not scaled up to a great extent, but we have been able to get full conversion of millimolar concentrations of different substrates. So I would believe that this is a good way forward. And I hope it answers.
I have a question, and it's not scientific, but one thing the hub is very keen on is supporting early career researchers. So if you had any advice to give someone starting out in research or in this field, what would it be?
Right. Things are changing so fast. And clearly, I was speaking about how we want to contribute to machine learning. And interestingly, I have to do this collaboratively because I don't know anything about computation. But I've always collaborated with people in the realm of computation through all my career. We've done a lot of work on enzyme dynamics, and this requires a lot of computation. So I've had tremendous support from colleagues. And so one recommendation is either learn computation or learn to collaborate. We can't do it all. So much of the work that we do is collaborative. All kinds of directions. You can't be an expert in everything. So become an expert in something that's useful. Know how, what questions do you want to ask? And if the best way to get the answer isn't within your scope of expertise, go out and get it, reach out, forge good, strong collaborations. Collaboration can be super long lasting. Some people I've been working with for so long, and also to foster student exchanges. If you can find, I don't know, for example, I'm going to throw a question back at you, Josie. Does the UK Catalysis Hub, does it finance exchanges?
It's potentially a political question because we have put some funding for early career research as secondments into the new grants. So it's a watch this space. And where we've been able to, we've done it in the past. We have students at the moment in Valencia.
It's such an interesting way to help promote really great research that you can't do alone in your silo. Yeah, I think that's those would be my recommendations.
We've not, we have. Oh, there's a thanks for the answer and a wonderful talk. The questions. That was the first question. We've not had any further questions in the chat, so I will say thank you again for an absolutely fascinating talk. It's not my topic, but I ripped it. Oh, Adrian just raised a hand. Let me see if I can. Adrian, you can unmute yourself.
Hi, Joel. Can you hear me okay?
I can. Hi, Adrian.
Lovely talk, beautiful work. Thank you.
Thank you.
Josie, I couldn't put my question in the chat. It was disabled for me. So, but yes, I think your question, Joelle, about can the hub support international collaborations? Well, one way or another, we would certainly like to do that in the next phase. And that includes, linking computation with experiment of various sorts. So I think that would be a great thing to do. And in terms of, linking the dynamics, so the data that you have is really rich, and we can learn computationally from that, as well as, I hope, simulations pinpointing that. So I guess the question I would have is, what is useful for you? How much is it useful to narrow the search space, and whereabouts would you like us to be targeting?
I don't know. And one of the reasons is cytochrome P450 BM3, or cytochrome P450s in general, they're so complex because, we chose to focus our mutagenesis efforts to a kind of a half dome above the heme. So that's okay, fair, because that's where the tunnels arrive into the active site, and the substrate's arriving, the product is leaving. But what about the electron transfer? And what about oxygen coming in and out? And there's so many other issues. What about just stability? So stability already, this is nicely complementary. If we can make predictions of amino acid substitutions that might be productive, but first screen them for stability and rule out some, that already is productive. But with respect to the actual reactivity, I think we're kind of too early. So we're trying, we're working now. We're starting a collaboration with Yuri Damborski and Carlos Acevedo Rocha using these data sets. And from a dynamics point of view, we have tried some pretty advanced methods, and we have not been able to identify any component of differing dynamics in the active site area that correlate with differences in reactivity we're observing.
Sure.
So, you know, I don't know.
It's a challenge, and I think there is no simple answer. I think, you know, one of the things you've nicely highlighted is the different dimensions that are important, and, you know, increasing one reactivity will maybe not help with another, or it might. I think, and something, you know, we might want to follow up on, there are sort of non-equilibrium approaches that could be useful for identifying hotspots and things, but we shouldn't go, you know, we can talk about that offline. I'm
Yeah,
conscious,
okay.
and Josie said this, so if anyone else is having the same issue as me and not able to type into the chat, you can, I think, raise your hand if you've got a question for Joelle. But otherwise, thanks very much. Lovely talk. And yeah, it'd be nice to talk more with you.
Thanks,
Nice
Adrian.
to see you. And congratulations on the ice hockey.
Oh, yes. Oh, yes.
That was a big win.
Oh, it was bigger than you can imagine.
I'm
For
sure.
something- For, and I can tell you 51 reasons.
Congratulations.
Okay. Well, no one else has said they can't use the Q&A. A couple of people have said they can't use the chat, so apologies for that. Hopefully you saw our messages.
I was using the wrong bit. Sorry, my fault.
It all worked out in the end. So thank you again, Joelle. It's been a very great pleasure to host you, and hopefully at some point in the future we can get you from person.
Thank you very much, everyone.